Tigon. A Tigon is the hybrid offspring of a male tiger and a female lion, created through deliberate captive breeding in zoos and research facilities. Unlike the more famous liger, Tigons are significantly smaller because the lioness carries genes that actively limit fetal growth during gestation. Scientists first produced Tigons in the 1800s to study interspecies fertility, genetic dominance, and reproductive compatibility between big cats. The result? A creature with faint tiger stripes, a compact muscular body, and noticeably reduced size compared to both parent species. Tigons rarely exist in the wild. They're bred exclusively in controlled environments for research purposes or exotic animal collections. Most are also sterile, unable to produce offspring themselves. Critics argue that hybrid breeding offers no conservation benefit whatsoever and prioritizes human spectacle over animal welfare, raising serious ethical concerns about forcing genetically incompatible species to reproduce purely for curiosity, entertainment, or profit alone. Geep. The Geep is a genetic chimera, part goat, part sheep, created by scientists at Cambridge in 1984 through embryo fusion. Researchers took cells from both species at the earliest developmental stage and physically merged them into a single organism, producing an animal with mixed tissue from two species coexisting in one body. The Geep had patches of wool and goat hair scattered across its skin, and its internal organs contained cells from both animals. A biological impossibility in nature, the experiment was designed to explore cellular compatibility and organ development across species boundaries. Geeps don't reproduce naturally. They exist only in laboratory settings. Enviropig. The Enviropig was a genetically modified pig developed at the University of Guelph in Canada, engineered to produce 30–65% less phosphorus pollution in its manure. Scientists inserted genes from mice and bacteria directly into pig embryos, allowing the animal to digest phosphorus far more efficiently than natural pigs. The goal was purely environmental, reducing toxic water contamination from large-scale industrial pig farming. Enviropigs were never approved for commercial sale, and the entire project was shut down in 2012 due to mounting regulatory concerns and widespread public opposition. Critics questioned whether altering an animal's genome to solve industrial farming problems was ethically justified, especially when the root issue is overproduction and factory farming systems themselves. Environmental groups argued the focus should be on reducing herd sizes, not redesigning animals. The Enviropig remains one of the most debated cases of agricultural genetic modification. Spider goat. Spider goats are real, genetically modified goats engineered to produce spider silk proteins in their milk. Scientists at Utah State University inserted genes from golden orb weaver spiders directly into goat embryos, enabling the animals to secrete silk proteins that can be harvested and spun into ultra-strong fibers. The experiment was funded by the military. Spider silk is stronger than steel by weight and lighter than Kevlar, making it incredibly valuable for bulletproof vests, parachute cords, and advanced medical sutures. The goats live on controlled research farms and appear completely normal, except their milk contains microscopic silk strands that can be extracted and processed into usable material. The goats themselves remain healthy and show no adverse physical effects from the genetic modification. The controversy? Critics argue that treating animals as biological factories for industrial material production reduces them to mere tools, raising serious questions about consent, animal welfare, and the ethical limits of genetic engineering for human profit. Featherless chicken. The featherless chicken was developed in 2002 by Israeli geneticist Avigdor Kahaner, bred specifically to survive in hot climates without air conditioning. Kahaner used selective breeding to create chickens with a genetic mutation that prevents feather growth, leaving them completely bare-skinned. The stated goal was to reduce heat stress in industrial poultry farming and lower energy costs. But the result was widely criticized. Featherless chickens are more vulnerable to parasites, skin damage, and cold shock. They also suffer from social rejection by other birds and cannot engage in natural preening behavior. Animal rights groups condemn the project, calling it an example of prioritizing profit over welfare. The experiment was halted. The featherless chicken remains a symbol of how far selective breeding can go wrong. Beltex sheep. Beltex sheep are a heavily muscled breed created in Belgium through decades of selective breeding, designed to produce maximum lean meat with minimal fat. Breeders amplified a natural mutation that causes extreme muscle growth, resulting in sheep with unnaturally thick hindquarters and sculpted bodies. The breeding wasn't accidental, it was deliberate genetic selection for commercial meat production. Beltex sheep are now raised across Europe and valued by the meat industry for high yields. However, their exaggerated musculature often leads to lambing difficulties, skeletal stress, and mobility issues. Critics argue that breeding animals purely for appearance and profit, while ignoring health consequences, is ethically unjustifiable. The Beltex remains controversial because it represents selective breeding taken to an extreme. Double-muscled whippet. The double-muscled whippet is a dog with a genetic mutation in the myostatin gene, a protein that normally limits muscle growth. When breeders in the racing industry discovered this mutation, some began selectively breeding for it to create faster, more muscular dogs. The result? Whippets with twice the muscle mass of normal dogs, bulging physiques, and increased sprint speed. However, the mutation often causes health problems, including heart strain, joint stress, and shortened lifespans. Double-muscled whippets are banned from many dog shows and discouraged by ethical breeders. The controversy centers on intentionally amplifying a genetic defect for competitive advantage. Critics call it a clear example of prioritizing performance over animal welfare. Saiga antelope. The saiga antelope has a bulbous, trunk-like nose that looks completely out of place on a hoofed mammal, evolved specifically to survive extreme temperature swings in Central Asian steppes. This oversized nasal structure filters dust, warms, freezing air in winter, and cools blood during summer, functioning like an internal climate control system. The nose contains complex internal chambers lined with mucous membranes that condition every breath before it reaches the lungs. Saigas can survive in temperatures ranging from 40 to plus 46, a range that would kill most antelopes. Found across Kazakhstan, Mongolia, and Russia, their population has crashed by over 95% since the 1990s due to poaching and disease. Their appearance challenges the standard antelope design entirely. These animals reveal a fundamental truth. Human biology is specialized for endurance and adaptability, but not extremes. We cannot survive venom, dehydration, boiling chemicals, or temperature swings the way these species do. Evolution shaped each creature to master a specific survival challenge humans would fail within hours. What seems impossible is simply refined biology pushed to its functional limit. Nature still holds secrets that challenge everything we know. If you are among the few who seek the raw, undocumented truth of our world, stay with us. There is much more to uncover.